Hybridoma technology. So it is used for producing the large number of identical antibodies. It is known as a monoclonal antibodies. Now, history of that, it was discovered by the G. Kohler and C. Milstein in 1975. For this achievement, they get the Nobel Prize in 1975, the branch of physiology and medicine. The hybrid cells are produced by fusing B lymphocyte with myeloma cell or tumor cell. So in this production of hybrid cell, we are fusing two cells. One cell is known as B lymphocyte and other is myeloma cell. Now this B lymphocyte have the ability to produce the number of antibodies, and this tumor cell have the have the indefinite growth. So both cell having its different activity. B cell produce antibodies and tumor cell or myeloma cell identify the growth. This is why two cells are fused for the production of hybrid cell. Due to its different activity, they are fused and we get the material what we need. Procedure of production of monoclonal antibodies is the mouse is immunized by the specific antigen injection against which monoclonal antibody have to be produced. Here we are done this process to isolating or selecting the mouse in which we are injecting the antigen. That antigen act against monoclonal antibody production. So here when we are introducing the antigen, immune system of mouse produce the monoclonal antibody. After 72 hours of immunization, spleen is collected from mouse. After 72 hours, spleen is collected from mouse because that spleen part producing the antibody of B cells. Now this B cell are fused with immortalized myeloma cell by PEG or Sendai virus. So here we are fusing this B cell with the myeloma cell by using PEG. B cell are fused with myeloma cell, and fifth is fused cells are incubated in the HAT medium. This HAT medium, it is hypoxanthine and aminopterin and thymide combination. Now hybridoma cell or fused cell are selected using selective media called as HAT medium. HAT contains these three components. The unfused B cell will die due to their short lifespan, and the myeloma cells can synthesize DNA nucleotide using two pathways. In first is the de novo pathway, second one is salvage pathway. So here in the production of monoclonal antibody, we are using HAT medium which contains hypoxanthine, aminopterin and thymidine. Now these are help to synthesize the DNA of nucleotide in myeloma cell. Now this HAT medium, the myeloma cells are unable to replicate because the de novo pathway is blocked by aminopterin in the medium. When de novo pathway is blocked, cell will utilize slavage as alternative pathway. So in short, myeloma cell are unable to replicate itself in de novo pathway because due to use of aminopterin, they are blocking its way. So when the de novo pathway is blocked, the second option is the salvage pathway. So myeloma cell utilize that salvage pathway, but it cannot take place due to the lack of HGPRT. It is hypoxanthine guanine phosphoribosyl transferase. So it is contributed by B cell and is rich in HGPRT. So this is the simple understanding that HAT medium having aminopterin. Myeloma cell are used for the production of hybrid cell. Now due to that aminopterin, myeloma cell does not synthesize itself. So we have the option is a salvage pathway, but this salvage pathway required HGPRT. Now myeloma cell having lack of that HGPRT. That's why we have to contribute this B cell because B cell is rich in HGPRT. The salvage pathway is also inhibited due to the mutation of thymidine kinase, an enzyme that catalyzes the phosphorylation reaction. The resulting clone of hybridoma cells secrete large quantity of monoclonal antibody. This is a representation of hybridization technology. In first, we are introducing the antigen into the mouse. Due to that, they are producing the antibodies in mouse, and then we are isolating spleen cells from that mouse, and side by we are isolating the myeloma cell which are having a TK factor, and the fusion of this both cell myeloma and B cell is done, and we get the hybrid cell. And after isolating and identification on the HAT medium, we get the monoclonal antibodies. Application of this monoclonal antibodies, so used for the detection of pregnancy, detection and treatment of the cancer, diagnosis of leprosy, treatment of the autoimmune disease. Radiolabel monoclonal antibody are used in vivo for detecting or localizing the tumor antigen, used for making immunotoxins that inhibit the protein synthesis. For example, Shigella, ricin, diphtheria toxins. This kind of immunotoxins are used. Now this is the simple representation in which epitopes antigen are given to the mouse. After isolation of the serum, we get the polyclonal antiserum, which is antibody 1, 2 and 3, 4. Next, after 72 hour isolated spleen cell. So after 72 hour we take the spleen cell of that mouse and isolating it. So after that isolation and combination of myeloma cell, we get the hybridized hybridomas, and then after select the clone, and due to that selection we get the different kind of antibodies. Application of antibodies, so they are improving they are provide its use into the diagnostic, imaging and therapeutic reagent in clinical research. Now these antibody are used as a diagnostic reagent and detect the pregnancy. Also they are detecting the pathogenic microbes, measure the blood level of the various drug and detect the antigen by certain tumors. By using hybridoma cell, or we can say monoclonal antibody, pregnancy test will done. A pregnant woman has the hormone human chorionic gonadotropin HCG in her urine. It means if the woman is not pregnant, this hormone will not found in her urine, but when she is pregnant, she contain the HCG in her urine. Now monoclonal antibodies to HCG have been produced, and these have been attached to enzyme which can later interact with a dye molecule and produce a color change. So here we are producing the monoclonal antibody which attached with HCG, and due to that the color change will produced and we get the result of pregnancy. Diagnosis of HIV. So the test of HIV infection is based on detecting the presence of HIV antibody in the patient's blood serum. So here it is used in the detection process of so many diseases.